Hey students, this lesson is all about how cells and multicellular organisms communicate with one another. We'll focus more on examples of cell-to-cell communication. In the subsequent lesson, we'll learn more about the mechanics of how cells respond to those signals. Here's a layout of our discussion. First, we'll learn how cells can communicate directly by cell-to-cell contact. Then we'll learn how cells can communicate over short distances through a process known as local signaling. We'll end by learning about a few examples of long-distance cell communication. Before we dive into the details, it's important to mention that there are three phases of cell signaling: reception, transduction, and response. Reception is the process by which a signal binds to a receptor either on the surface of the receiving cell or a receiver that's in the cell's plasma membrane. Transduction is a series of steps that involves the passing of the original message through the cell through a series of relay proteins and secondary messengers. Response often involves the activation or deactivation of a particular gene. So reception, transduction, and response. All right, on to our discussion of communication via cell-to-cell contact. This happens two ways: cell junctions between cells or cell-to-cell recognition via membrane proteins. Animal cells sometimes communicate by sending electrical impulses from one cell to a cell neighbor directly next to it. This is different than neurons. We'll talk more about neurons later. So gap junctions are protein channels on a cell's plasma membrane through which an electrical signal can pass from one cell to the next. So here you can see the plasma membrane of two cells that are sandwiched together. The gap junction is a protein channel that connects the two cells. Electrical impulses can travel through these gap junctions. Here's another visualization of gap junctions, this time in cardiac muscle. So we have a muscle cell here, a muscle cell here. This is the plasma membrane of each cell kind of not fused, but sandwiched together. And then these gap junctions are protein channels through which an electrical signal can travel. Gap junctions are important for the contraction of heart muscle cells and therefore for the pumping of blood throughout the body. Electrical signals arise in your heart in something known as the SA node or the sinoatrial node. That electrical impulse is propagated through the cardiac cells through gap junctions, causing them to contract. So the SA node makes this electrical signal and then it passes through the heart cells via the gap junctions. In a similar manner, adjacent plant cells can communicate with one another when a chemical messenger synthesized by one cell passes to a neighboring cell via a channel in the cell wall known as a plasmodesma or plasmodesmata for plural. So you don't have to know the details of how all of this works, but it's important to know that one plant cell can communicate with its neighbors by synthesizing a protein messenger that then passes to a neighboring cell through the plasmodesmata. And this actually happens when one cell in a plant can trigger the other cells to flower. Yet another example of communication by cell-to-cell contact is cell-to-cell recognition. One example of this is in the way immune cells recognize and destroy pathogens. Antigen-presenting cells are white blood cells that phagocytize or engulf foreign cells. So this is an antigen-presenting cell, this is an antigen-presenting cell, kind of the tealish cell in this picture. The foreign cells have non-self proteins on their surface that your antigen-presenting cells don't recognize. These non-self proteins are known as antigens. Once an antigen-presenting cell phagocytizes a pathogen, it will break down the pathogen into its little components, and it will display some of the pieces of the pathogen or the antigens on the outside of the antigen-presenting cell. Antigen-presenting cells have a molecule known as MHC on their surface. They use MHC to show antigens to other cells, almost like a way of saying, hey, look at what I just found and I destroyed it. MHC is like a trophy case that all antigen-presenting cells have on their surface. When an antigen-presenting cell phagocytizes a pathogen, the antigen-presenting cell shows off pieces of the pathogen, known as antigens, in the MHC trophy case. So here's our antigen-presenting cell. It's showing pieces of the pathogen it just engulfed, and it's showing it to other cells by holding it out on its MHC2. Helper T cells and killer T cells, which are other types of white blood cells, recognize antigens only when they're displayed on the antigen-presenting cell's surface via an MHC trophy case. The T cells will use their T cell receptor to bind to the antigen that's displayed on the MHC2. So they'll hook together on this antigen, but only if it's attached to an MHC2, and it will become activated. This activated T cell then helps out with something known as the adaptive immune response. Now let's talk about local signaling, which is when cells release chemicals that travel across short distances to influence cells in the nearby vicinity. We'll discuss 4 types: neurotransmitters, plant immune response proteins, proteins involved in quorum sensing, and proteins known as morphogens that can influence embryonic development. Neurotransmitters are chemicals that travel from the space at the beginning of one neuron, the empty space, to the next neuron. This space between the 2 neurons is called a synapse. The story here is that an electrical signal travels down the length of one neuron. It gets converted into a chemical signal known as a neurotransmitter. The neurotransmitters then diffuse across the synapse, and then those neurotransmitters can either cause or suppress a response, cause or suppress an electrical signal in the postsynaptic cell. The electrical signal itself cannot cross the synapse, so that's why neurotransmitters are so handy. In order to cause a response in the postsynaptic cell, the neurotransmitters must bind to protein receptors on the surface of the receiving cell. Plant immune responses are another example of local signaling. Plant cells can recognize pathogens that require living cells in order to thrive. When a plant cell is invaded by such a pathogen, it secretes a chemical known as salicylic acid, and then it kills itself through programmed cell death. In plants, this programmed cell death is known as the hypersensitive response, or HR response for short. The secreted salicylic acid triggers a hypersensitive response in nearby plant cells. This creates a ring of dead cells around the pathogen, so the pathogen can't spread. So you can see here, the pathogen probably invaded these center cells, and then those center cells secreted salicylic acid, killed themselves, and then the surrounding cells also completed the HR response and killed themselves as well. That way the pathogen cannot spread to the healthy cells. We've already discussed the type of local signaling known as quorum sensing in a previous presentation. Refer back to Cell Communication Part 1 for more information on quorum sensing. Quorum sensing is used by bacteria in your mouth to secrete a layer of slime or biofilm, and they can use that biofilm to attach to the surface of your teeth and then form colonies of bacteria, which can give rise to cavities and to oral disease. Another example of local signaling are proteins known as morphogens that can cause changes during embryonic development. The dorsal lip, which is found on the outer edge of a developing embryo, is known as the organizer because it secretes chemicals known as WNT or Wnt proteins. These Wnt proteins diffuse across the embryo, causing changes that induce the formation of the dorsal axis in an embryo. So the dorsal axis refers to your belly and your back. Your dorsal side would be where your backbone is, or where the dorsal fin of a shark would be, and then your ventral side is your belly side. In this experiment pictured here, scientists removed the dorsal lip from one frog embryo and transplanted it onto another frog embryo. So the receiving frog embryo had 2 dorsal lips. Both dorsal lips synthesized the Wnt proteins, and this caused the frog to develop 2 dorsal axes. So the resulting tadpole formed 2 backs. So you can see here that the dorsal lip synthesized Wnt proteins here and here, so it starts to form a back on both sides, so we ended up with this tadpole. Finally, we have long-distance signaling. 2 examples of this are your endocrine and nervous systems. Today we'll focus on hormones, which are part of your endocrine function. The hormone oxytocin is secreted by the pituitary gland on your brain, and it's released into the bloodstream. As blood carrying oxytocin travels past uterine cells, the oxytocin binds to oxytocin receptors on the uterus, and this causes the uterine cells to contract, helping to push a baby through the birth canal. Oxytocin also triggers lactation, or the production of milk in new mothers. So that's all, folks. Rewind and review as you need. Thanks for watching.